Hello everyone and welcome to our channel, Mootechie. A warm welcome to all the dairy technologists and students joining us today. We're going to take a detailed look at the fermentation process, a cornerstone of industrial microbiology. Our presentation is titled simply, Fermentation Process, which introduces our main topic. Let's begin with a definition. Fermentation is a chemical process where molecules like glucose are broken down without oxygen, a condition known as anaerobically. More broadly, the term originates from the foaming seen during wine and beer production, a practice that dates back at least 10,000 years. This frothing is caused by the release of carbon dioxide gas, though this wasn't understood until the 17th century. The French chemist Louis Pasteur, in the 19th century, provided a more specific definition. He used the term to describe the changes caused by yeasts and other microorganisms growing in the absence of air. He also recognized that ethyl alcohol and carbon dioxide are not the only possible products. Next, we explore the anaerobic breakdown of molecules. A key discovery in the 1920s revealed that extracts from muscle tissue could convert glucose into lactate without air. Crucially, the same intermediate compounds produced in grain fermentation were also found in muscle tissue. This led to an important generalization. Fermentation is not unique to yeast but is a common process in many instances of glucose utilization. This brings us to glycolysis. Defined around 1930, glycolysis is the metabolic breakdown of sugar into lactate. It is a form of fermentation where the 6-carbon sugar glucose is split into 2 molecules of a 3-carbon acid called pyruvic acid. This process is coupled with the transfer of chemical energy to synthesize ATP or adenosine triphosphate. The pyruvate can then be oxidized in the presence of oxygen or reduced without oxygen to products like lactic acid or alcohol. This pathway is known as the Embden-Meyerhof pathway. So the term fermentation now refers to an enzyme-catalyzed, energy-yielding pathway in cells that involves the anaerobic breakdown of molecules like glucose. The necessary enzymes are located in the cytoplasm. These reactions are universal, occurring in muscle, yeast, bacteria, and plants. This is summarized by the chemical equation, where 1 molecule of glucose yields 2 molecules of ethanol and 2 molecules of carbon dioxide. Now let's define fermentation technology. It is any process that produces a product by the mass culture of a microorganism. Biochemically, it is an energy generation process where organic compounds act as both electron donors and acceptors. Industrial fermentation processes begin with suitable microorganisms under controlled conditions. The products are diverse, including alcohol and glycerol from yeast, solvents and acids from bacteria, and acids and vitamins from molds. Ethyl alcohol from starch or sugar is also an important biofuel. Let's now examine the chronological development of the fermentation industry through a detailed table. This table outlines 3 main stages from before 1900 to the present. It tracks the evolution of main products, from alcohol and vinegar to antibiotics and organic acids. Vessel technology progressed from wooden barrels to modern steel fermenters. Process control advanced from simple thermometers to computerized systems using sterilizable electrodes. Culture methods evolved from simple batch to fed batch and continuous systems. Quality control became very important. Pilot plants became common, and strain selection moved from pure cultures to sophisticated mutation programs. Another important table lists major recombinant proteins used as therapeutic agents. It groups them by function, such as blood factors, hormones, and cytokines. For each, it shows the protein name, the production system, like E. coli or mammalian cells, and its clinical use. For instance, insulin is produced in E. coli and yeast for diabetes treatment, and erythropoietin is made in mammalian cells to treat anemia. Now we'll look at types of fermentation processes based on the product. The main categories are microbial cells for biomass like baker's yeast and single-cell protein, and microbial enzymes such as amylase and protease. These enzymes are produced commercially from various sources and have the enormous advantage of being producible in large quantities via established fermentation techniques. Other important process types involve microbial metabolites, which are split into primary and secondary metabolites. We also have recombinant products like interferon and vaccines, and transformation processes, such as those used to produce vinegar. Microbial metabolites are specifically divided into 2 groups, primary metabolites and secondary metabolites. Let's examine a table of primary metabolites and their commercial significance. Ethanol is used in beverages and as a biofuel. Citric acid has various food industry uses. Glutamic acid is a flavor enhancer. Lysine is a feed supplement. Nucleotides also enhance flavor. Phenylalanine is a precursor for the sweetener aspartame. Polysaccharides are used in food and for enhanced oil recovery. Vitamins are important feed supplements. We also have a diagram showing how primary metabolites give rise to a variety of cell substances. This complex chart illustrates the metabolic pathways, starting from central molecules like glucose and pyruvate, branching out to form nucleotides, amino acids, lipids, and other essential cellular components. It visually demonstrates the interconnectedness of metabolism. Now here is a table for secondary metabolites. Penicillin and streptomycin are antibiotics. Bleomycin is an anti-cancer agent. Lovastatin lowers cholesterol. Cyclosporine A is an immunosuppressant. And ivermectins are antiparasitic agents. A corresponding diagram illustrates the production of secondary metabolites. It shows pathways starting from pyruvate and acetyl-CoA, leading to the synthesis of diverse compounds like polyketides, terpenes, sterols, and antibiotics. This highlights the specialized nature of secondary metabolism. The relationship between biomass, nutrients, and metabolite concentration is shown in a graph. It depicts the tropophase, where growth occurs, followed by the idiophase, where secondary metabolites are produced. The biomass increases until a limiting nutrient causes it to plateau, which often triggers the production of the secondary metabolite. Recombinant products represent a major advancement. Recombinant DNA technology allows genes from higher organisms to be inserted into microbial cells like E. coli or yeast, enabling them to produce heterologous proteins. Animal cells are also used in fermentation systems, though they present challenges like fragility and low cell density compared to microbes. Products from genetically engineered organisms are numerous and include interferon, insulin, human serum albumin, and various growth factors. Key factors in process design include ensuring the product is secreted, minimizing its degradation, controlling when synthesis starts, and maximizing the expression of the foreign gene. The parts of a fermentation process can be divided into 6 basic components. First, formulating the growth media. Second, sterilizing the medium and equipment. Third, producing an active pure culture for inoculation. Fourth, growing the organism in the production fermenter under optimum conditions. Fifth, extracting and purifying the product. And sixth, disposing of the waste generated. A schematic diagram represents a typical fermentation process. This flowchart shows the progression from stock culture to shake flask, then to a seed fermenter, and finally into the main production fermenter. The medium is formulated and sterilized beforehand. After fermentation, the culture fluid is processed, biomass can be harvested, and the product is extracted from the cell-free supernatant, purified, and packaged. The diagram also includes effluent treatment, emphasizing waste management. There are 2 main types of fermentation processes based on the physical state: solid-state fermentation and liquid-state fermentation, also called submerged culture. Solid-state fermentation, or SSF, is also known as surface fermentation. It involves microbial growth on solid particles without free water, where growth and product formation occur on the substrate surface. Substrates like rice bran are saturated with water and inoculated with microbes. It's ideal for fungi and is more cost-effective due to smaller vessels and lower water usage. SSF has many applications. It can produce high-value products like enzymes and antibiotics. Notably, nearly a third of industrial enzymes in Japan are made this way. It's also used to produce organic acids and ethanol from starchy substrates and to improve the digestibility of fibrous materials for animal feed. Liquid-state fermentation, or submerged culture, is performed in large tanks. It is ideal for bacteria and yeast. The main methods within LSF are batch fermentation, continuous fermentation, and fed batch fermentation. Batch culture involves culturing cells in a vessel with an initial charge of medium that is not altered during the process. It is simple but results in continuously changing conditions, making it an unsteady state system. A typical batch growth curve is shown. It starts with a lug phase, where cells adapt to the environment. This is followed by exponential growth. Then growth slows, just so, a deceleration phase, leading to a stationary phase where growth stops due to nutrient exhaustion. Finally, there is a death phase. During the lug phase, cells are metabolically active and adapting. This is followed by exponential growth at the maximum possible rate. However, exponential growth is limited. As nutrients deplete, growth decelerates and enters the stationary phase. The final death phase involves cell lysis, and most processes are stopped before this point. Batch fermentation has advantages. It has a reduced risk of contamination due to the short growth period. There is lower capital investment and more flexibility with different products. It also allows for high raw material conversion. Disadvantages of batch culture include lower productivity due to downtime for cleaning and sterilization. It requires more instrumentation checks and incurs higher costs for preparing subcultures. Labor costs are higher, and there are greater hygiene risks. Continuous culture involves continuously adding fresh nutrients and removing spent medium and cells. This allows a time-independent steady state to be achieved, which is excellent for studying microbial behavior. In a mixed bioreactor, this can be done as a chemostat or a turbidostat. In a chemostat, growth is controlled by limiting the concentration of one nutrient, maintaining a constant chemical environment. In a turbidostat, the feed rate is adjusted based on turbidity to maintain a constant cell concentration. The chemostat keeps chemistry constant, while the turbidostat keeps biomass constant. A chemostat operates by connecting a growth chamber to a sterile medium reservoir. Fresh medium is added at a rate that limits growth by controlling the supply of one essential nutrient. An overflow drain keeps the volume constant. This system prevents the conditions that normally cause the stationary phase. A diagram shows a simple laboratory fermenter for continuous cultivation. It includes features like air filters, pumps for medium addition, a culture vessel, and a product receiver, illustrating the continuous flow of materials. Fed batch processes were devised early for yeast production. They involve an initial batch period, followed by the controlled addition of nutrients without removing culture fluid. This keeps growth limited by the substrate, preventing the production of undesirable byproducts like ethanol, and allowing for high cell densities. In a fed batch, cells are grown in batch mode initially. Then, a substrate solution is fed into the reactor to maintain a specific growth rate and minimize inhibitory byproducts. The volume in the reactor increases over time. Fed batch is useful for achieving high product concentrations. For growth-associated products, the growth phase is extended. For non-growth-associated products, cells are grown to a high concentration first and then induced for production. This is especially useful for recombinant systems. Advantages of fed batch include higher yields from a defined cultivation period. It allows optimization of conditions for specific growth phases and is nearly stationary, which is good for unstable cultures. Disadvantages include lower productivity due to reactor downtime and higher costs for process control. Finally, we have a short assessment quiz. First, what is a primary metabolite? Please provide an example. Second, what is a secondary metabolite? Again with an example. Third, is a batch process a steady state process? And fourth, in a turbidostat, is the cell concentration constant? Thank you for joining this comprehensive journey through the fermentation process. We have covered everything from its basic biochemical principles to the various types of industrial processes, their control, and their vast applications. This knowledge is fundamental for anyone in dairy technology or related fields. We hope you found this session incredibly informative.